Avrobio is an ex vivo lenti gene therapy company. We founded ourselves 5 3/4 years ago, so a little bit older than homology. And everything we do applies the ex vivo lenti platform to lysosomal storage disorders. And I guess if I summarize where we are, we have 2 waves of gene therapies. The wave that is in the clinic, wave 1, which is Fabry disease, Gaucher disease, and cystinosis. And we've treated 19 or 20 patients, so when we get the slides, I'll be able to share the clinical data with you. And we have a second wave of gene therapy assets that have finished preclinical development. All of the CMC work is more or less complete, and we're just filing INDs and CTAs to be in the clinic no later than this time next year. So the second wave is Pompe disease, infantile and late onset, Hunter disease, and Gaucher disease type 3. Any progress on the slides? So I'll just wing it. If I try to sum up the company, the mission is to free patients from a lifetime of disease. What that means in our world is A, to untether the patient from biweekly infusions of enzyme, but also more than that. And in addition to just getting the patients untethered from enzyme, we want to halt disease progression. Enzyme replacement therapy is the standard of care today, and it was a godsend 20 years ago. It does a pretty good job slowing down the rate of decline. But the whole premise of Avrobio and the raison d'etre of gene therapy is, what if you could deliver 24/7 expression of enzyme? And the real unmet need, the deficiency of today's standard of care, centers around the pharmacokinetics of ERT. It's the right enzyme, bad pharmacokinetics. And so this is what we're trying to do. What if you could bathe patients' cells, tissues, organs with enzyme 24/7? So I'll just advance. Forward-looking statements will happen. I refer you to our SEC filing. And this is us really on a snapshot. So I mentioned that we're in LSDs. I think what we're really proud of is that we've spent the early years of the company rolling up what is now a leading platform. And a few brag points is we're the first gene therapy in the clinic in Fabry. We're the first gene therapy in the clinic in Gaucher disease, the first gene therapy in the clinic in cystinosis. And we will be the first lentiviral approach in our second wave of indications. And many people know these indications from the Shires and Genzymes. These were really some of the foundational assets of those great biotech companies. And even today, $5 billion of revenue is generated from the founding companies. We'll talk a lot about Plato. When you hear about AVRO, our platform, we've spent a lot of time trying to take an open Petri dish science approach and turn it into a pharmaceutical-grade medicine. And I'll talk a little bit about that at the end. On the right-hand side of the screen is where we are. As I mentioned, we've treated 18, 19 patients. We're very happy to say that at this point in time, every patient, every blood draw, every clinical trial, we're seeing the kind of efficacy, and now it's becoming durable efficacy as some patients are out 2, 3+ years with no waning of effect. The energy units at AVRO today are being massively applied to regulatory filings. And we expect in 2022 to initiate multiple phase 3 registration trials in 2 or 3 separate indications. So for us, that's a big step forward in terms of going to focusing on getting into the clinic to now beginning to focus on getting out of the clinic. Here we go. So here's the pipeline. I think I've covered that. I'll skip that. I did mention the $5 billion of revenue, but there is an additional important point, and that's that these indications still today cost the healthcare system a fortune. Today's standard of care direct costs are hundreds of thousands of dollars a year, not even factoring in indirect costs every year for life. So there's an Erasmus study that shows the lifetime cost in Fabry disease is $12 to $14 million. And of course, one of the big questions at this conference and in our field in general is health policy reimbursement. And I would say that at least we're blessed with indications that if we hit our target product profile And if we demonstrate a safe, effective, one-time therapy, we should be able to show a really compelling health economic advantage, even just limiting it to the 5-year cost offset. Talked about the kinetics. Now, in our world, in order to be first-line therapy, we have to not only show efficacy and durability, but we, of course, want to show safety. And lentiviral gene therapy has been demonstrated to have a pretty excellent safety profile over 2,000-3,000 patients that have been treated to date. We calculate about 500 in monogenic rare disease, but we also include the CAR-T experience, of course, if you're looking at untoward safety events. And we can say in our data to date, we've seen no AEs and no SAEs related to the gene therapy, and our heavy focus is on how do we continue to improve the patient experience related to the concomitant conditioning regimen. And what you're seeing on the screen here, on the left, is a seminal paper from Geoff from MSK in New York, who really finally pooled all of the transplant data around the world. And what he uncovered was that there really was this Goldilocks mid-range dose of the conditioning agent busulfan, which if you hit it, you could get the chimerism, get the engraftment of these gene-modified cells that you would like, but you avoid the out-of-range toxicity. So that's job one. The target exposure rate has been identified, but it's really tough to hit it because there's tremendous inter- and intra-patient variability. So if you dose 10 patients, you're going to get 10 different exposure rates. We've tried to solve that by doing a really meticulous area under the curve dosing where we break the dose into 4 mini doses over 4 days, multiple blood draws to make sure that we don't only target what is called Bu90, but we hit Bu90. And by doing so, we've been able to generate the adverse event profile that you see on the right-hand side of the screen. So I don't want to trivialize those because, of course, they are adverse events, but by really optimizing this approach and by targeting indications where the patients come into the equation with healthy immune systems, healthy liver, and most importantly, healthy bone marrow, we think that we can harness the busulfan conditioning to the type of adverse events that we've seen in our early patients, where the adverse events are transient, meaning they arrive at about the beginning of week 2 and they depart at the end of week 2, and they're highly predictable. And our belief is that if we can continue down this path, we do have a compelling value proposition relative to the benefit that accrues to these patients. So now to jump into the data, I really only have time to cherry pick a few data pieces for each of the 3 clinical stage indications, but this is perhaps our most compelling data that we've generated to date. This is our phase 2 program in Fabry disease. We've completed our 5-patient phase 1. We now are about 3/4 of the way through a global phase 2 trial, and the primary endpoint is a kidney substrate biopsy, the average number of inclusions per peritubular capillary. So the patient has a kidney biopsy at baseline and then again at 1 year, and after a single infusion of gene therapy, what you see is what a nephrologist would equate to full effective clearance. Net a 93% reduction of substrate in the kidney. So we're, of course, very pleased with that. There are products that have been approved with lower percent reductions than that. We do anticipate that this will be the primary endpoint in the phase 3 study that we hope to be able to initiate this time next year. And then, again, I won't have time to cover all of the data, but giving a quick summary of our phase 1 data, these are patients that for many, many years were on enzyme replacement therapy. They stopped briefly, received gene therapy, were put back on enzyme replacement therapy, and then gradually the ERT was titrated off. And so what we can say is that now going out 3.5 years, all patients that received the gene therapy are doing well. All patients that had ERT discontinued remain off ERT. And the gene therapy not only controlled, for example, on this slide, the lyso-GB3, the toxic metabolite in the plasma, but controlled it at a level better than baseline ERT, 25% lower than what the patients within patient control ERT was able to generate. And the key point that we see very clearly visually is that there's no waning of effect over time. So we're very pleased with the data so far. Jumping to a second LSD, cystinosis often is a less well-known, less well-understood lysosomal disorder. And unlike our other 5, it's not an enzyme deficiency, it's a transporter protein. There's about 2,000 patients in the world. And again, I don't have time to show all the data, but what I'd like to show is that in the first patient, what we were able to look at baseline and then at 1 year was the average volume and number of crystals that form in the cells of these patients via a skin biopsy and a rectal biopsy. And what you see is a substantial reduction, 35% and 76%, which is, of course, important because it highlights that the gene therapy is doing something and is likely distributed through the body, but there are more relevant functional measures, and one of them is the eye. This is a devastating disease, and there are 90% of these kids have a kidney transplant, but they also have severe visual impairment. And what you're looking at in blue across the top is actually the patient's cornea, and the white are the crystals that accumulate, and they accumulate so profoundly that they overlap like you're looking at a cloudy sky. And the patients not only have to take 30 pills a day of cysteamine, but they also have to take eye drops of cysteamine every few hours. So in this patient, all pills, all eye drops were discontinued, and when you look 1 year later, not only is there an improvement, but there's a marked improvement. So the crystals haven't 100% cleared, but they very, very materially cleared, such that the patient scored better on a photophobia scale and certainly can resume activities such as leaving the house without dark glasses. So an interesting parameter for us. And I'll close cystinosis with another finding that we really didn't anticipate. And this slide tends to remind us when we're designing our trials in gene therapy in general that we have to really understand the mechanism of action because, of course, evolution made us such that proteins are multifunctional, and it just turned out that the cystinosin protein not only results in those catastrophic effects to the eye and to the kidney and other areas, but it also is associated with the synthesis of melanin. And cystinosis patients tend to present like this young gentleman on the left, and after even a few short months after a single infusion of gene therapy and complete discontinuation of cystinosis, his entire visual appearance began to change. And he himself said for the first time in his life, he looks like his brother because he comes from a pigmented family. So I'm saying this not just for sort of the show business aspect of it, but if you jump back to Fabry for a minute, we're looking very narrowly at enzyme up, substrate down. We should invite ourselves to think about what does really the effective gene augmentation do to these patients, and it could have effects that we're not properly thinking about or measuring. The third and final clinical stage program is Gaucher disease type 1. Very analogous to Fabry disease. Instead of kidney and cardiac, it tends to be liver, spleen, bone. But like the others, it is a head-to-toe disease with head-to-toe manifestations. So I'll share our first patient. This is a 32-year-old Gaucher disease type 1 female who was on the Arthur's old VPRIV product from Shire from the age of 3 until 32. And what you're looking at is one of the key biomarkers, chitotriosidase. And what you see at the beginning of this curve is that VPRIV was doing an okay job controlling chitotriosidase, but because this patient is a human being, inevitably she went through a period of non-compliance. She was missing her ERT infusions, and chitotriosidase spiked from 150 up to almost 3,000. So now if you jump ahead and look at that very same patient where, again, VPRIV was discontinued in our study, but rather than the chitotriosidase on the left jumping from 150 to 3,000, it was actually significantly reduced, down 44% or 48%, and lyso-Gb1 also reduced. So we're very satisfied. It's an N of 1 in its early data, but certainly as we produce more, if we can continue to generate this kind of impact, we'd be very pleased. Now, just to close, I mentioned our platform. We call it Plato, and we have been heavily focused on trying to take this open Petri dish science manufacturing process and turn it into a pharmaceutical-grade medicine with a heavy focus on safety, but also on manufacturability, on scalability. And I don't have time to go through our entire process, but I would just highlight a few things that we're particularly happy are in place today. One of the things that we've invested in from the founding of the company was automation. And we believe that it's necessary if you want to develop a scalable, global product. And so all of our gene therapies are manufactured in this roboticized, closed system. And of course, that provides a lot of benefit. Certainly scale, it increases tenfold right away because you can have rows of these automated pods in one low-grade clean room, but it also allows you to globalize because even today, we manufacture in Melbourne and Houston and California and are opening up in Europe. And really, the benefit of having a closed automated system is the ease with which you can expand from one CMO to the other. You still need data, you still need engineering runs, you still need to submit, but there's a more feasible path than were it not to be a closed system. A few other things, we manufacture in a serum-free suspension culture 200-liter bioreactor with a 10 to the 9th titer. It means every single run can produce enough vector for 40 or 50 patients. So with today's infrastructure, we can do easy math to get to thousands of patients a year. We've optimized our vector. We began with an academic vector, and now we believe we have a state-of-the-art 4-plasmid lentiviral vector system, and we've optimized our approach to conditioning. So we define Plato as best in class, and the job never ends. We're continuing to work on how do we continue to optimize it to get to the point that we really have a viable commercial stage platform. So I hope that I've imparted our excitement at the progress that we've made since I was last able to present to you. Our clinical data continues to hold, and our platform is in place. Thank you very much.